McChicken. Where'd my thing go? Oh, all right. So question 1, or A, says identify the pigment chlorophyll or bacteriorhodopsin used to generate the absorption spectrum for each of the graphs. Explain and justify your answer. So graph 1 and graph 2, we need to decide whether we're looking at one for bacteriorhodopsin or for chlorophyll. Remember, when we were talking about this, it said that chlorophyll was green. Oh, that's not green. That's blue. That's green.
I thought you were trying to spell green.
So chlorophyll is green, and we said that bacteriorhodopsin, I'm just going to put Bact, was purple. Okay, so Bact was purple. If I look at these graphs, what are these graphs showing me? So the wavelength of light reflected or absorbed?
Absorbed.
So on my x-axis, it's telling me the wavelength in nanometers. On my y-axis, it's telling me my relative absorbance, meaning how much actually gets absorbed at each wavelength. Remember, if something is green, is it absorbing green light?
No.
It's actually reflecting it. All right? So right here, it gives me this little chart right here on the bottom, gives me all the different colors and what is their respective wavelength of light. So chlorophyll, if it were green, it's saying things that are green are the wavelength of light 495 to 570. That's going to be the wavelength for green. Okay? And things that are purple or violet are going to be 380 to 450. Or bluish should be 450 to 475. So we'll start with chlorophyll here. Which one of these two do you think has to do with chlorophyll? So if it is green, is it going to be absorbing a lot of green light?
No.
It would actually be reflecting it. And so right here it says that green is going to be 495 to 570. If I look on graph 1 from 495 to 570, did it absorb a lot of wavelength of light? It did. Meaning this one's not chlorophyll. Okay? Meaning if I look right here, 475 to 570, so like right here-ish, didn't really absorb anything. Meaning this one's going to be chlorophyll. Okay? Because it absorbed a lot in the like 440 spectrum. It absorbed a lot in the like 680 spectrum. 680 ring a bell? Think about the light-dependent reaction. Photosystem 2, that very first photosystem, it had a P680, meaning it loves the wavelength of light 680 nanometers. This one right here for chlorophyll absorbs a lot of light at that 680 nanometers wavelength. But it doesn't absorb much at 475, and it doesn't absorb much until it hits about 550, 575. All right? So that's what's going on there. Meaning, graph 1 is going to be for which organism? That's the bacteriorhodopsin. So I'm just going to put Bact. Okay? Hold on, folks at home. My phone is ringing. Giraffes. It's going to be right next to the track. So it's the concession stand. All right, perfect. Thank you. You almost killed me. Your bag. All right. So we know graph 2 is going to be chlorophyll because if I look at the relative absorbance, things that are green are actually green because they're reflecting it, meaning it would not absorb any of that light. It's not absorbing anything in that green wavelength, so it's going to appear green. This one right here is reflecting a lot at this thing, so it's reflecting a lot in here, that's absorbing a lot in there, and meaning it's reflecting a lot right here-ish. So from like 350 to 500 range, and if I look, 350 to 500 range is violet and blue, or purple, which is that color. That's why this one goes for, the graph 1 goes for bacteriorhodopsin, which if you read the preface right here, it says chlorophyll A is commonly found in green plants. Other pigment bacteriorhodopsin is found in purple plants, or purple photosynthetic bacteria. Okay? That's why it's purple, because it's not absorbing the purple, it's absorbing the other stuff. So it's absorbing the green, it's absorbing the red. Right here it says for B, in an experiment, identical organisms containing a pigment from graph 2, so we're going to only look at graph 2, as the predominant light capturing pigment is separated in 3 groups. Organisms in each group are illuminated in a single wavelength of light. So it's going to look at one for an, wrong thing, I didn't want that one. It's going to look at an organism for 650 nanometers. So we're going to hit it with 650 nanometers. We're going to hit it with 550 nanometers. And we're going to hit it with 430 nanometers. It wants to know which one's going to do it the fastest. So we'll say first or fastest. Which one's going to be the middle rate for photosynthesis. And which one's going to be the slowest for photosynthesis. Okay, so we've got to decide that 650, 550, or 430. We've got to predict that and justify why, looking at this graph. So remember, we were looking at 650, 550, and 430. So based off these 3 wavelengths of light that we have, so we've got one wavelength about right here, we've got one wavelength right here, and we've got one wavelength right here. Which one is do you think will do the photosynthesis at the fastest rate? Because it absorbs the most sunlight. This 430 will, because if you look at 430, 430 is right here. It's absorbing a ton of light right there. So our first one right here would be 430 nanometers, because it has a very, very high amount of absorption at that wavelength of light. Our second highest one, which one do you think it would be? 650 or 550? It'd be 650, because right there, it hits right here on here. So second would be 650, because there's still a higher absorption level than the other one. And then our third and final one is going to be 550 nanometers. Because almost everything's reflected at that wavelength, so it's not really going to absorb anything. That's why conceptually, if I were to take a green light and try to grow a plant under there, would it work? It wouldn't, because it doesn't like that. When we do a photosynthesis lab, we're going to try taking a plant, we're going to measure the rate of photosynthesis when exposing it to green light. We're going to see what happens. And we're also going to expose it to blue light. We're also going to expose it to red light, see what happens. Okay? Any questions on any of these? None?